Hi friends, today we are going to see the past year NEET questions for the chapter Biological Classification, and today in this video we will cover part 3. So earlier we have completed 2 parts, so today this is part 3. So let's begin with the first question here. The chief advantage of encystment to an amoeba is dash. So what is encystment? First we'll discuss that. Encystment can be defined as the process by which many organisms adopt the dormant and highly resistant stage of the cyst. See, sometimes if you have studied about the protozoans, there are 2 stages of infections. Sometimes certain organisms are infectious in their cyst stage, or sometimes they are infectious in their flagella stage. So that depends. Now this encystent is actually an advantage for an organism. Okay, so here it will become resistant. It is actually a dormant stage where there is not any much activities, but still it is active inside. It is not showing any action, but still it is having life, preceding the release of the reproductive stage. So you're given 4 options here, right? So encystment enables the zygote to survive under adverse or unfavorable conditions. Thereby it helps in the survival of the organism. Okay, so what is encystment? It is a protection. Okay, it is a protection actually for the organism, and it saves from adverse conditions. So therefore the correct answer to this question is option B. So here you have to your right side a picture which shows about encystment. Okay, the circular one, the circular notion here, it shows encystment. Okay, fine, let's move to the next question here. Which bacteria is utilized in Khobar gas plant? It's again an easy question for you because we already discussed about methanogens in the earlier parts. We talked about the waste, or after the metabolic activity, the waste that is eliminated from the ruminants, especially the cows, cattle. Okay, they all come under that. Their metabolic waste which comes out are rich in methanogens. Okay, so the correct answer here is methanogens. Methanobacillus occurs in the marshes and also in dung. It produces methane gas under anaerobic conditions and utilized in Khobar gas plant. So I have given you a picture which represents that. So the ruminants, as I said, they are the major production of this methanogens. So here you have the cycle. From ruminants it will produces methanogens, and the bacteria is methanobacillus, and from there it produces methane, and that is under anaerobic conditions. So that is what we use in the Khobar gas. Therefore the correct answer is option A. Next question here, which was asked in the year 2002. Which statement is correct for bacterial transduction? So before we go into the options given here, we need to discuss what is transduction. What do you mean by transduction? See, transduction is a process by which a foreign DNA will be introduced. Okay, a foreign DNA will be introduced, by which a foreign DNA is introduced into a bacterial cell by a virus or a viral vector. So what is it? Introduction of a foreign DNA. Okay, introduction of foreign DNA into a bacterial cell by a virus or a viral vector. An example is a viral transfer of DNA from one bacterium to another, and hence this is an example of gene transfer, and this is called as horizontal gene transfer. So what is the answer now? Now I think you can guess the answer what it is. Transduction is a virus mediated gene transfer in bacteria. Therefore the correct answer is option A, transfer of some genes from one bacteria to another bacteria through virus. Virus will be the vector there. Therefore the correct answer is option A. Next question here. What is true about Cyanobacteria? We already studied what is Cyanobacteria. What is the other name for Cyanobacteria? It is also called as blue-green algae, right? Also known as Cyanophyta. It belongs to the phylum consisting of free-living photosynthetic bacteria, and they are endosymbiotic. What do you mean by endosymbiotic? Of course they are symbiotic, but there are 2 categories, exo and endo. Endo means within the organism we are talking about, not outside. Endosymbiotic plastids, a sister group of Cyanobacteria, are present in some of the eukaryotes. Apart from that, what else you can explain about cyanobacteria? What do they do to the humans? Exposure to cyanotoxins caused by drinking or swimming in contaminated water or breathing air containing cyanobacteria or other toxins, they are said to affect skin. Okay? Sometimes they even affect your nervous system. If it is inhaled, or it is been if you start drinking contaminated water and it's in a high quantity, then it seems to infect your nervous system and sometimes even your liver. A wide range of symptoms can develop from the cyanotoxin exposure, including skin rashes and skin irritation. So what is true about cyanobacteria here is, the correct answer is option A. It is oxygenic and it has with, or it is presents with, nitrogenase. Cyanobacteria are oxygenic phototroph. Many members perform nitrogen fixation. Therefore, the correct answer is option A. Next question here. What is true about Archaebacteria? Okay. So you have you're given 4 options here. All are halophiles, all are photosynthetic, all are fossils. See, out of the 4 options given here, Archaebacteria are the most ancient and the halogenous group of bacteria that are called as living fossils. So here, all are fossils. If you ask me, it is not all are not fossils, all are not photosynthetic, and all are not halophiles. As I already said, you have acidophiles, depending upon the source that they live in. Certain bacteria can live on rich salt conditions or rich acid conditions, hot springs, temperature variations. So there are conditions like that. The correct answer is option D. They are the oldest of the living beings. Okay, they are even considered now as a living fossil because they have evolved a long back ago. Okay, the correct answer is option D. Next question here. The main difference between the gram-positive and gram-negative bacteria resides in their dash. Okay, let me explain you an interesting thing here. When we talk about what is gram-positive and gram-negative, gram-positive takes in purple stain. Okay, it takes in purple stain, while gram-negative takes in the pink stain. Okay, it takes in the pink stain. What is this stain? Have you ever thought about? See, we do a lot of research on these microorganisms because they are the one which cause a lot of problems to us, and they are the one which cause a lot of infections and diseases in especially human, who are the highest of all living organisms. So we do a lot of research. Okay, so what we can do is, here it depends upon the stain. See, simply if we have to view a bacteria, or simply if we have to view about any kind of organization, their internal structure, whatever it might be, we require a special methodology so that we can view it under the microscope. That is why they use something called staining technique there. So when you do a staining technique there, they correctly depict one thing. Gram-positive bacteria will take a purple stain, while gram-negative bacteria will be taken by the gram-negative bacteria will take the pink stain. So that itself will show the variation. So now I have given you a clear message about what is gram-positive and gram-negative. So here, as I said, gram-positive and gram-negative. See, even though they all belong to a common group of bacteria, they are taking in different stains. So that possibility is because of the cell wall. Okay, we all know the cell wall or the cell membrane, whatever we talk about, anything to cross in should cross over the cell wall only and go inside. And prokaryotes, I said, they have a special cell wall which will protect them and help them in the survival. So this cell wall is very important. This cell wall variation only counteracts with the stain, and one will take one purple and the other will take pink. So it is because of the cell wall. All this explanation and why I am giving here is, it is very it's clearly understood here. The stains, the bacteria is able to differentiate between different stains. It is able to differentiate the composition of the stain. Okay, so that variation is because of the cell wall it has. The correct answer is option A. Okay, the bacteria which retain the blue or purple color after staining will be called as gram positive, while the other one will be called as gram negative. Okay, next question here. Photosynthetic bacteria, okay, they have pigments. Okay, they have pigments in dash. This is a very easy question for you, I think. Fine. Chloroplast, chromoplast and leucoplast are the type of plastids which are found in higher plants. Okay, leucoplast, chromoplast and chloroplast, they are found in higher plants. Of course, they are all plastids, they all contribute to the colors, the pigments. Okay, so photosynthetic bacteria have pigments from chromatophores. Chromatophores contain colored pigments, while leucoplasts contribute to the colorless pigment. This is colorless. Okay, chloroplast contributes to green color, chromoplast contributes to, they contain colored pigments. It's a combination. Likewise, photosynthetic bacteria have pigments from chromatophores. As I already mentioned, these three, chloroplast, leucoplast and chromoplast, are the ones which are present in higher organisms, while bacteria, when we come to a lower living organism, chromatophores is the main thing. Actually, they don't have anything in particular. They have a combination of everything. When we say chromo, it's a combination of colors or pigments. So that is present in the bacteria so that it can attain the color. Okay, therefore the correct answer is option D. Okay, next question here. In prokaryotes, the genetic material is dash. I think this is a very easy question for you to answer. See, as we all know, prokaryotes, though they are the lower living organisms, they have a single cell and still they have a complex structure to study about. Here, if you see in prokaryotes, the genetic material will be circular and they will have the presence of histones. Okay, in prokaryotes, nucleoid consists of double-stranded circular DNA with histone protein. Therefore, the correct answer is option D. This is a very easy question. I didn't give much explanation about this because we already discussed in the earlier parts. Okay, next question here. A few organisms are known to grow and multiply at temperatures of 100 to 105 degrees Celsius. They belong to dash. So, this is again an easy question for you to answer. So, when we go to 100, 105 degrees Celsius and all, what kind of bacteria they could be? They are the ones which can survive in extreme conditions, right? It's understood. They are the ones which can survive in extreme conditions. They are Archaebacteria. So, we already studied about it in the earlier parts where we spoke about Archaebacteria. They can survive at any condition. While Eubacteria is a true bacteria, they require normal temperature to survive, while Archaebacteria, they can survive in extreme temperatures. Okay, so they have a special cell wall, and that is not made up of peptidoglycan but made up of fatty acids. That is a major advantage for them to survive such conditions. That is one thing you need to keep in mind. Therefore, the correct answer to this question is option A, marine Archaebacteria. Okay, next question here. Transfer of genetic information from one bacterium to another in the transduction process is through dash. So already we discussed what is transduction in the earlier question. It is the transfer of a foreign DNA into a bacteria, and where you can use a virus as a vector. So here you have four options here, right? So transfer of genetic information from one bacterium. You're transferring a gene, okay, from one bacterium, okay, from one bacterium to another. So it can happen by physical contact. When it happens through a physical contact, you call that process as conjugation. That's something which will happen naturally. But when you are transferring manually or artificially, you will call that as transduction. So what is the right answer here? Bacteriophages release from one donor bacterial strain. Therefore, the correct answer is option B, where you have a picture to your right side which shows a picture of bacteriophage there. Okay, the correct answer is option B. So this is how a bacteriophage structure will look. Okay, next question here. The hereditary material present in the bacterium E. coli is dash. Hereditary means it will come from ages to ages. It's a continuation from generations to generations. Okay, so what is this E. coli? You have to let's see about it. It's a gram-negative bacteria. Okay, it takes in the pink stain, and sometimes it is said to cause some intestinal infections. They are a natural possessor of your intestine, but sometimes they might also cause certain infections related to intestine especially. Apart from that, the hereditary material present in the bacterium E. coli is double-stranded DNA. The correct answer is option C, and to your right side you can see the picture of E. coli, where it is where the full body is full of cilia, and it has flagella to one side, and it is lophotrichous. It is understood. So this is how when it is stained and you have a 3D image of it. Okay, usually it will be simply rods in pink color. It will be like this when you view it under microscope. So this is actually a 3D view that they have given you. Okay, next question here. Sex factor in bacteria is dash. This is again an easy question for you. What do you mean by, so what is the sex factor that is present in bacteria? Bacteria, as you all know, it is a microorganism, and it is said to be advantageous and disadvantage in many categories. Now, what is the sex factor of bacteria? As I already said, what does a bacteria do? See, the fertility factor, which was termed as F by one of its discoveries, I mean, who discovered it? If you see, it is Esther. Who discovered it? Esther. Okay, so that is called as fertility factor. You call it as F factor or F replicon. So this is the F factor which is present in most of the bacteria, and especially E. coli, it is very active. The sex factor is also called as F replicon, allows the genes to be transferred from one bacterium carrying the factor to another bacterium, and this will lack by the factor conjugation. So apart from that, the fertility factors will also allow the genes to be transferred from one bacterium to another, and the F factor is carried from the, so from where it will be carried, if you see, it will be carried from the F episome. The process begins from here. Okay, therefore the correct answer is option B, F replicon. Bacteria results in high frequency conjugation. It allows the bacteria to produce sex pilots necessary for conjugation. Okay, next question here. Temperature tolerance of thermal blue algae is due to dash. As I already said, there is a particular when we talk about extreme condition survival bacteria or microorganisms, it is understood they have something unique in them. See, sometimes, let's take an example here. When you are in a class, imagine every month you get cold, then you are less immune, it is understood. And there are people who don't get cold itself. They have good immunity, they can resist infections. So this talks about the survival here. This scenario can be compared here also. Temperature tolerance of thermal blue-green algae is due to dash. Some of the blue-green algae can tolerate extreme of temperatures due to the presence of gelatinous sheath. You call it as gelatinous sheath. Okay, and its compactness of protein molecules that are present exclusively in the protoplasm. So now I've given you the answer. The correct answer is option D, homopolar bonds in their proteins. Homo means same, so you find the repetition of the bonds there. Okay, next question here. Nitrogen fixer soil organisms belong to dash. So this is an easy question for you to answer. Yes, it is bacteria. Only some bacteria and blue-green algae, especially Cyanobacteria they are called as, have the capacity to fix atmospheric nitrogen. Bacteria, I can give you examples, like here you have examples: Acetobacter, Rhizobium, Frankia, etc. Some of the examples of bacteria who can fix atmospheric nitrogen. And blue-green algae, you have Nostoc, Oscillatoria, Anabaena, and there are some more organisms which can fix atmospheric nitrogen. Therefore, the correct answer is option B, bacteria. So this question was asked in the year 1994. So next question here, Entamoeba coli causes dash. So this is again an easy question. Before that, Entamoeba coli is a protozoan. Okay, it's a protozoan. So what does Entamoeba coli causes? See, I'll discuss about each option so that you will understand. Pyorrhea is caused by bacterial infections along with other factors, while diarrhea is caused by rotavirus, and dysentery, if you see, it is caused by Entamoeba histolytica. So here they have asked for Entamoeba coli. Of course, it causes some intestinal infections, but out of the 4 options given here, none matches the Entamoeba coli infection. Therefore, the correct answer is D, none. Okay. So here you have the Entamoeba coli cyst morphology. So here if you see in Entamoeba coli, the infectious stage is the cyst stage. Okay, easy question. The correct answer is option D, none, because all the 3 go for different other responsible factors. Hence the answer is D. With this we complete the part 3 of biological classification. Hope you understood. So we will meet in the next part of the same chapter. Thank you.